A protein is a biological molecule made of amino acids. Proteins have many different functions based on their shape. They can defend, transport, provide structure, or be contractiles. They can also act as enzyme, hormones, storage, or receptors. What they do depends on their structure. Form fits function. As mentioned before, proteins are made of amino acids, and there are 20 different types of them. An amino acid is made of the amine group, NH3, and the carboxyl group, COOH. Connecting them is a carbon atom. Also connected to the carbon atom is a hydrogen on one side, and the side channel, also known as the R group, on the other side. There are 20 different R groups, which is why there are 20 different amino acids. The backbone of the amino acid is everything except the R group and is made of many polar covalent bonds. Whether an amino acid is polar, nonpolar, or positively/negatively charged is determined by the R group. The primary structure is the sequence or order of amino acids, which is encoded within DNA. Connecting the amino acids are the peptide bonds, which are covalent bonds formed by a dehydration reaction. The secondary structure is when the polypeptide backbone forms hydrogen bonds with itself. Specifically, the carboxyl group of one amino acid forms a hydrogen bond with the amine group of another amino acid. As the polypeptide collapses, alpha helices, which look like loops, and beta pleated sheets, which look like zigzags, form.
The tertiary structure is when the polypeptide chain forms a three-dimensional structure due to interactions between the R groups of amino acids that make up proteins. 4 R group interactions include covalent bonds, ionic bonds, hydrogen bonds, and hydrophobic interactions. Covalent bonds form disulfide bridges between amino acids that have sulfur in their side chains. Ionic bonds form between two opposite electrically charged amino acids, positive and negative charges. Hydrogen bonds form between a partially positive hydrogen and a partially negative oxygen or nitrogen in the side chains. Lastly, hydrophobic interactions are when side chains with nonpolar and hydrophobic R groups fold together inward to shelter from water through London dispersion forces. Quaternary structures are proteins made up of many folded polypeptides called subunits. Not all proteins have 4 levels, but some do. Our protein is an enzyme which helps to speed up or catalyze biochemical reactions that happen in our cells and helps to sustain life.